Dr. Thomas Ciula, ophthalmology medical lead from CEPAC Therapeutics. Thank you very much. Thank you for coming to Kansas City to be with us today. We appreciate it.
Well, first of all, thank you very much for having me speak today. As you mentioned, my name is Dr. Thomas Ciula, and I'm the Ophthalmology Medical Strategy Lead for CEPAC Therapeutics. I'm a board-certified ophthalmologist and retina specialist. I co-directed the Retina Service and Ocular Angiogenesis Research Laboratory at Indiana University School of Medicine, and I remain a volunteer clinical professor of ophthalmology. I trained in the Harvard system, served numerous roles in over 100 national clinical trials, and have co-authored over 130 Medline Index peer-reviewed papers. I also hold an MBA and have become interested in linking my expertise in retinal disease and research with the development of novel therapies such as Luxturna that serve unmet needs. So in the next 4.5 minutes, I'm going to stress 3 points. Number one, prior to the availability of Luxturna, nearly all patients with RPE mutation-associated retinal dystrophy would have progressed to complete blindness as there were no treatment options. CEPAC wants to ensure that all eligible patients have access to this novel treatment. Therefore, CEPAC has priced Luxturna responsibly and has created outcomes-based payer rebate arrangements that recognize its longer-term efficacy, and also has explored alternative payment options that would allow for installment payments. Number two, CEPAC also has an interest in ensuring ICER's report on Luxturna does not limit patients' access to the therapy because of a premature base case economic model that makes headlines. As CEPAC has stated throughout the ICER process, the model was created too early. There are limited data available to incorporate into a model, and I think that's just been reflected. Restrictions on data have been necessary as CEPAC was undergoing FDA review until December 19, 2017, and is still collecting utility data. Nevertheless, CEPAC has worked with ICER throughout the process to improve the model and provide suggestions for the most appropriate data to include. Number three, and most importantly, Luxturna addresses a dire unmet need in a very small patient population which is impacted by ultra-rare genetic disease. This warrants appropriate inputs to determine cost effectiveness and an appropriate cost effectiveness threshold. When ICER includes some or all of these inputs as sensitivity analysis to the base case model, ICER's own report demonstrates that Luxturna is often cost effective for many patients. However, these analyses are obscured by the premature base case model that makes headlines. Let me briefly address some of these inputs and cost effectiveness thresholds individually. First, ICER's base case continues to underestimate the treatment effect from Luxturna and uses health utilities that are inappropriate for this population, as we just briefly touched on. Specifically, when ICER employs health utilities that reflect the rapid decline in health as patients progress to blindness, Luxturna appears to be cost-effective under the modified societal perspective using standard cost-effectiveness thresholds. This is shown in ICER report table 4.9. Second, even when ICER fails to use this more appropriate utility function but does incorporate both a lifetime treatment effect and indirect costs into its model, as we just touched upon, and as CEPAC recommends, Luxturna is indeed cost-effective at the current list price at all cost-effectiveness thresholds for a 3-year-old patient and at a cost-effectiveness threshold of $250,000 per quality for a 15-year-old patient. This is shown in ICER's own report, table 4.8. Third and finally, ICER recognizes that other benefits and contextual considerations must be accounted for when reviewing an ultra-orphan drug. It therefore reports benchmark prices for higher cost-effectiveness thresholds. These are more appropriate for an ultra-orphan drug like Luxturna versus more traditional drugs with larger patient populations and multiple treatment options. Therefore, CEPAC requests that ICER more clearly explain that when indirect costs are taken into account, as we just touched upon, even in the base case scenario, Luxturna is indeed cost-effective for a 3-year-old patient at the standard $150,000 per quality threshold, and for a 15-year-old patient at a $500,000 per quality threshold. So in summary, number one, CEPAC is addressing any potential concerns about cost directly with payers through its novel payer programs, including its outcome-based rebate arrangements that stand behind the durability of Luxturna, and potential alternative payment options that would allow for installment payments. Number two, it is disappointing that the ICER press release emphasizes only the premature base case economic model. And finally, number three, ICER's very own report demonstrates that Luxturna is indeed cost-effective for many patients when it uses appropriate health utilities, uses indirect costs that address the high societal impact of blindness, as we heard today, considers a lifetime treatment effect, especially in the absence of data suggesting otherwise, and/or uses higher cost per quality thresholds for innovative therapies addressing ultra-rare disorders, such as this one that leads to total blindness. Unfortunately, payers who do not review the report carefully could easily miss these points and inappropriately and unfortunately limit access to sight-saving therapy for those in need. Thank you very much.
Thank you very much for your comments. So we thought if you're available, we would love to ask some questions and I think build off of the comments that you just provided. And I think
Could I back up just one about the MLMT? Sure. So there were some comments about the MLMT. So first of all, I want to emphasize that it's a novel endpoint. People ask, what does it mean? Well, it turns out that it is very much grounded in reality. If you look at ICER report, I think it was on page 19, those light levels correspond to real-world living conditions. So in other words, you'll notice that the average patient improved from 50 lux to 4 lux. So they could function at 50 lux at an outdoor train station and at 4 lux at an outdoor parking lot at night. So it actually is grounded in reality. It was an endpoint that Spark developed in conjunction with the FDA. The FDA was very, very interested in a functional vision versus visual function. So I would state that the MLMT is indeed grounded in reality. It is a real-world test. That's what it was designed for. That's what the FDA wanted, a functional vision test.
I think that I appreciate that remark and that clarification. I think we heard, kind of commentary and testimony from from Caitlin and Janet that affirmed that particular position too.
Another comment I'd like to clarify. So there's a lot of confusion in this investigational agent arena with RP65. There's been 2 other studies using different vectors. And using those different therapies, they didn't achieve the durability effect that we've achieved. Unfortunately, the results that Spark has, and for which it got approval, those results are being lumped in with these other therapies. And I just want to emphasize that the therapies are totally different. You could actually have the exact same gene being augmented with the exact same vector, but they're very different therapies still. And they're different because, for example, the Spark product removes empty capsids. Without going into all the technical detail, by removing vectors that don't contain the gene, you actually enhance the efficacy because you can saturate the receptors with empty capsids, and that decreases the therapeutic efficacy. We also add surfactant to the product. Surfactant minimizes the vector from sticking to contact surfaces as it's drawn up and it goes through tubings and is injected onto the retina. We have a different promoter than some of these other therapies. So to lump this therapy in with the others is not scientifically accurate. And our data so far shows that there is durability. And I think in the absence of the contrary, I think we have to assume that there is durability, as is meant to be with any genetic therapy.
And my guess is we'll get a question from the group also on maybe some additional comments on durability. But Rachel, would you like to ask a question first?
Sure. Thank you for your remarks. I had a question about efforts to identify patients early. We've heard how important it is for patient outcomes and quality of life to find patients and treat them early, but we've also heard from Dr. Kay and Dr. Bankin about some of the structural difficulties in identifying these patients early. I'm wondering if there are any efforts underway from Spark directly to help clinicians find these patients earlier on, or if it's merely having physicians now with greater urgency know that this treatment is available.
Thanks for the question. So the question was, what sort of efforts are underway, maybe by Spark, but also the retina societies in general, to identify patients earlier? And so Spark had a program called ID or IRD, It was a free gene testing panel, 31 genes, including this gene. And it rolled out, I think, approximately a year and a half ago. It was actually greeted with great enthusiasm by the retina community. Unfortunately, genetic testing up until now has not been something that the retina community has adopted because up until now there's been no therapy for it, and often it's not reimbursed by insurance. So I think Spark's ID or IRD program has certainly increased awareness of genetic testing. It's becoming more and more commonplace. Dr. Kay can speak to it. She is an inherited retinal disease specialist, so she's obviously in tune to it. But I know in my interactions with more general retina specialists, they are now thinking about genetic testing. So I believe patients will be tested earlier as awareness increases.
Stephen, did you have a remark for me? Oh, there's Stephen.
So I'm David Rand.
Wait, I'm sorry.
From ICER. I'm intrigued by the argument about durability, that we have 3 years of published data, and therefore our assumption should be that something lasts a lifetime. What we've assumed is, with 3 years of data, that it lasts 10 years, continues to work for 10 more years. So we're giving 20 years of benefit to a therapy that's shown 3 years of benefit. Is it really your contention that a reasonable decision would be to say, we have 3 years of data, therefore it lasts forever?
Well, that's the whole, I can't speak specifically about, my crystal ball has cracks in it, but I can tell you that's the whole theory behind gene therapies. You add a gene, and that's constitutively expressed, and in the absence of data suggesting there's a waning effect, it's hard to assume otherwise, because that's the very reason for doing the gene therapy. I also want to address, at a very theoretical, high level, this concept of persistent degeneration. So we've heard that there are misfolded proteins being elaborated, and that contributes to degeneration. And that may be, but as Dr. Kay mentioned, most of the mutations are thought to be null mutations. In other words, there's a mutation that tells the cellular transcriptional and translation machinery to stop. And so the proteins aren't even manufactured. Another theory is that the reason for the degeneration is that there's a buildup of toxins. So if you break the cycle, if the RPE65 gene is missing and you break the cycle, that product in the cycle right before the gene is built up, and that's called retinal esters. So these retinal esters continue to build up, and they're thought to be toxic. By restoring the cycle, by actually metabolizing those retinal esters, you actually prevent the buildup. So theoretically, it may stop degeneration, and that's apparently what we've seen in early stage dogs. So, I think up until now we have no data with our therapy that there's a waning effect. I think you showed the curves, and if not, they're publicly available curves showing stable benefit up until 3 years in the phase 3 population, and up until 4 years in the phase 1 population.
I'm gonna move us to Rob's question, and then Reem, sorry.
Thanks. So I appreciate your comments about the MLMT as an effort to provide a real-world measure and context to the impact of the treatment. At the same time, another really important real-world impact would be quality of life data. I mean, I would think from what I saw there that most adequate measures of quality of life would basically scream with positive results based on what we saw from the MLMT. Now, there's a lot of talk in the report that data is available, or it was collected, but it has not been made available. Could you speak to why that is?
Well, thank you, actually, for answering that question, because it's a point that needs clarification. So the question is, what about our quality of life data? Well, as many of you know, there's this VFQ-25 quality of life tool. It's been validated. It's used in clinical trials. Unfortunately, it's geared towards adults, and it's geared towards adults that don't have very profound loss of vision. There are questions, for example, about driving on that quality of life survey, and that wouldn't be appropriate. So that survey was modified for our trial, and it was really modified towards functional vision and not so much quality of life. When that was analyzed, there was a statistically significant benefit, but it hasn't been validated because it has been modified, and it really focused on functional vision as opposed to quality of life. Spark is still collecting utilities, still doing a lot of work on this, and I think we'll have more answers in the future. But I think at the FDA AdCom, we heard stories from some of the patients, as well as some of the physicians, talking about throwing their canes away, being able to walk through crosswalks more confidently, going back to school, going back to work, and so forth. We also had Dr. Leroy spoke about his patients who have had efficacy for, I believe it was up to 10 years. But the question's a good one, and we'll have more data soon.
Thanks. Reem.
You know, this is very exciting, and the results are exciting, but I want to go back to the issue around durability when there is so much uncertainty around the pathophysiology and so many different mutations. And what we heard from the experts and from you guys, we're treating 20% of the retina only. We're not really treating the whole retina. So to me, I'm not an ophthalmologist, but from a clinician and from an epidemiologist standpoint, I do see why there would be some uncertainty that this is durable for life. It's different than if we're treating the whole retina or if we're 100% sure about the pathophysiology forever, and we know that there's no folding. There's a lot of uncertainty there that makes me question the issue with durability, yet I am super excited about the results.
That's an excellent question. I'm glad you asked it. It's actually a two-part question. So one, how much of the retina is treated? And so it's estimated between 20 and 30% of the retina is treated. But I can tell you as a retina specialist, we don't use most of our retinas. So if you were to just with your own hand, if you were to check your own side vision, I can see out to here. And out to here with this eye. So you have almost 150 degrees of field. And glaucoma patients actually do quite well with loss of side vision until it comes close to the center. So that much of the peripheral retina we don't use. And obviously the central retina is what's important. So as long as you have macular function and perimacular function, you can actually function quite well. The other part of the question about durability, I think we can agree to disagree, but I also think there are other issues in the model. So it's not just about plugging the durability in and looking at cost effectiveness. It's the other issues, looking at the nonlinear health utility. From your very own report, suggests if you use that nonlinear health utility that accounts for the rapid loss in health as patients progress to blindness, just that one factor alone makes this cost effective. If you look at from a societal perspective, that enhances the cost effectiveness. In fact, we had one mom who testified at the FDA AdCom. She indicates that she lost $1 million in her own income while she had to care for her blind child. And also, I think in this, as I mentioned, in this ultra rare orphan disease, we don't think the standard quality cost effectiveness benchmarks apply, because there's a lot of intangibles that are difficult to account in these models. And I think some of that's come out today in talking to Dr. Kay and some of our patients who are here to discuss that.
Do we have any questions from the ICER staff? Any additional, or no?
Well, thank you very much.
Anything? Okay, great. Aaron? Okay, thank you very much. We appreciate your time. So that sort of wraps up our morning session. I'm going to kind of pass it back to Steve for or to Sonia for some instructions for the next part of our day. But I appreciate the great dialogue today from everybody. Thank you so much for your participation in the morning session.
So I have the fun announcement, which is it's time for lunch, and we'll have lunch available for everyone in the audience right outside these doors, and for the panel we'll be where we were this morning. And we'll reconvene at 1, so we'll give you a little bit of extra time. Thanks.
Thank you.